I'm Paul Haider, a PhD student in the NeuroTeam A group at the University of Bern. And I will present to you today Latent Equilibrium, our unifying framework for arbitrarily fast computation and learning with arbitrarily slow neurons, which is, of course, a bold title, but let me explain to you what we mean by that. So the topic of this talk is efficient information processing in dynamical physical systems. What's that supposed to mean? So we mean systems that really implement the information processing with the physics and the dynamics of its constituents. And probably the best example of such a system is, of course, the human brain, which motivated a still ongoing endeavor to both understand information processing in the human brain and also to build bio-inspired hardware in order to replicate its function, such as with the spiky chip you can see on the right-hand side here, or all the other newer chips we've seen so far at MNIST conference. And now one of the key challenges is, of course, to understand learning in such systems, which can be translated to the credit assignment problem. And a solution that works quite well in the case of artificial neural networks is, of course, the arrow backpropagation algorithm. However, since backpropagation itself is not biologically plausible, there exists a lot of bioplausible approximation to this algorithm. So, for example, equilibrium propagation, or the segregated dendrites model, or the dendritic cortical microcircuits by Sacramento. And now we argue that all these bioplausible approximations, they share a common, rather fundamental problem that applies to any physical system, namely that their speed of information processing is, of course, fundamentally limited by the speed of their components, which for neural networks are, of course, these slow components are, of course, the neurons themselves. And because of their slowness, as we're going to see, all of these models of approximate backprop, they require some sort of relaxation. But yeah, to understand that, let's first have a closer look at what problems this intrinsic slowness induces. And therefore, consider this chain of two neurons the simplest possible feed-forward system. And now if we model them as leaky integrators and provide them with an input rate in blue that contains fast components or even jumps, and by fast I mean now in particular faster than the neuronal dynamics, which in the case of neurons are limited by the membrane time constant, then the membrane voltage is described by the leaky integrator dynamics, that is by this differential equation on the right-hand side, resulting in a both attenuated and also delayed version of the input signal. And likewise, in the case of output rates, which are usually some nonlinear function of the membrane voltage, there are significant differences than from what one would expect from an instantaneous system. And now in addition, this gets worse with every layer, such that in hierarchical networks, this the effective relaxation time is even a multiple of this membrane time constant. And this is also a well-known fact in biology, namely that cortical neurons need, of course, time to integrate their input with the characteristic time scale given by the membrane time constant. And therefore also networks in cortex need a certain relaxation time before all the neurons have access to what one would call the correct information. However, this is only the first part of this problem. There's a second problem, and that's even worse, and concerns learning in such slow dynamical systems. And in order to see that, let's use again a neural net that already does what it's supposed to do. So we use the same setup as before, just zoomed into the step input. And now without plasticity in the limit of vanishing learning rates, the purple membrane voltage relaxes towards this black dotted target. However, in the case of continuous learning during relaxation, plasticity usually tries to adapt the synaptic weights, as you can see in dashed in the lower plot, in order to minimize the difference between voltage and target, which first leads to an overshoot during the early relaxation, which then again needs to be undone during the late relaxation, as you can see dashed in the upper plot. And now there are two things to note here, because first, apparently the system adapts the weights even in perfectly trained networks, but more importantly, the synaptic weights, they do not simply revert to their previous state. Rather, the network somehow unlearns what it had learned before. But yeah, the field is of course well aware of this problem, and there are two apparent solutions that have been used so far to overcome this problem. The first one is, of course, to wait for equilibrium and use small learning rates in combination with long presentation times. And the other one is phase plasticity, meaning that plasticity is only active in the relaxed state, which is, of course, rather difficult in practice and also still slow. Okay, so far we've seen that, yeah, during relaxation, not only can the neurons be perceived as doing something wrong, but more importantly, also the slowness induces these timing mismatches between instructive signals and the neural activity, which makes it even more difficult to assign credit in such systems of slow components. And therefore, it's probably fair to say that this is an essential issue for information processing in neuronal substrates, not only in biology, but also in biologically inspired neuromorphic hardware. And now, to overcome the problems I just illustrated, we propose a framework for fast computation and learning in physical systems with slow components, which is based on four rather simple axioms. The first one is prospective coding, which should not be confused with predictive coding, because by prospective, we mean that the neurons, they look into the future using a linear combination of their membrane voltage and its derivative to overcome the low-pass filtering of the membrane. And as you can see, we denote this prospective voltage by U breve. And now, of course, the idea of taking the derivative into consideration is well known from control theory and has also appeared in the neuroscience literature before. So just to give you an intuition, already in the Hodgkin-Huxley mechanism, it is indeed the case that whether the neuron spikes or not depends on how fast the membrane potential is changing. But there's also direct evidence by Köngen, for example, of this prospectiveness in biological tissue, showing that the firing rate can indeed be phase advanced with respect to their input. Okay, so we've seen so far that this prospective voltage determines our neuronal outputs and can be seen as a prediction of the membrane after relaxation. And now, based on this variable, we define our prospective energy function. And that energy function consists of a mismatch term that somehow compares the output, or in other words, it compares what each neuron predicts it will be doing to what the presynaptic partners tell it to do, plus some output loss that measures the performance of the network under some task. And now the nice thing about this theory is that everything else follows from this energy function. So the third ingredient, namely the neuronal dynamics and morphology, are derived from a stationary energy function. Because requiring stationarity with respect to this prospective voltage, one obtains again the leaky integrator dynamics, well known from biology, where the individual terms represent the different compartments of the neurons. So you have the leaky soma, illustrated by this red triangle, which integrates both the input signals in the basal dendrites, as well as the error signals in the apical dendrites. And yeah, this derivation is in contrast to previous work that introduced neuron dynamics via gradient descent on an energy function, such as, for example, equilibrium propagation. And we argue that this difference is indeed crucial to solve the relaxation problem, because in our case, for a given input, our network moves by construction within a constant energy manifold. Or in other words, the membrane voltages, they may change all the time, but there's something that's always in equilibrium, which is also why we refer to our model as latent equilibrium. And now in hierarchical networks, these equations instantiate also a variant of error backpropagation, where we avoid the separate backward path by allowing the errors to influence the neuronal activity. And I will show you a detailed biophysical implementation of these equations in a minute. But first, let me show you that this equation does not only describe the bioplausible neuronal dynamics and morphology, but also implicitly addresses the relaxation problem that I introduced you to earlier. So in order to see that, on the left-hand side, I've shown you again the same plot that I showed you before, with the leaky integrator dynamics, where both the membrane and output rates are rather slow and not able to process the quickly changing input signal. But within our framework, that is, if you combine the leaky integrator dynamics with the prospective neuronal outputs, we can see on the right-hand side that we can again process information quasi-instantaneously and independent of how fast the input changes. And that although the membrane voltage depicted in orange is never in equilibrium. Okay, and now the last ingredient to our framework is synaptic plasticity, which we introduce via stochastic gradient descent on our energy function to obtain a local learning rule that relies only on information available at the locus of the synapse. And note that, yeah, that it's a local learning rule, although the locality was never an ingredient from the start. And this is also a variant of a powerful synaptic learning rule that has been proposed some time ago by Urbanchik and Sen. All right. Okay. Now, note that that by design here, the neuron and synapse dynamics are decoupled, such that for constant input, as I already said, the neuron dynamics move by definition within a constant energy manifold. And at the same time, then the synaptic plasticity then pulls down on this entire manifold, solving the relaxation problem for both inference and learning. And this is illustrated on the right-hand side, namely that within our framework, as you can see in orange, the energy jumps with the input, but then remains constant as long as the neuron dynamics evolve. And this, in contrast, this does not hold if you define the neuron dynamics via, for example, gradient descent on your energy, because then plasticity follows incorrect gradients during the relaxation. Okay, and now what we think is particularly appealing about this framework is that we have only a few premises, but a lot of conclusions. So we use a single simple prospective energy function to jointly derive the dendritic morphology, the neuronal dynamics, and the local synaptic plasticity and network structure from these simple principles. And this theory now unifies, in that sense, neuronal dynamics and learning. And only from this unifying view, it allows us to understand why a framework is defined or postulated in that way. Because we have the prospective coding that enables the instantaneous activity propagation, and then plasticity can be continuously active without disrupting the learning performance. Okay, but now enough theory. Let's have a look at some of the results. So first, to demonstrate learning in a network where the neurons are much slower than the signals they need to process, we did a simple Fourier synthesis task where we use a network that is driven by sinusoidal inputs of equidistant frequencies to approximate a target signal, in that case, a sawtooth signal. And now in this setup, the corresponding input frequencies are much bigger than the inverse membrane time constant, such that the neurons themselves are in principle too slow to represent these fast dynamics. But as you can see on the right-hand side, continuous plasticity still allows our model to approximate the target well. And that, yeah, although the membrane voltage is never in equilibrium. Then, of course, we also tested our model on standard machine learning benchmarks, such as the MNIST dataset, using a fully connected network with 2 hidden layers. And now here in gray, you can see the baseline that we obtained using vanilla backpropagation with stochastic gradient descent. Now in purple, you can see what happens in the case of a neural net with leaky integrator neurons. But apparently, because of the relaxation problem, no successful learning is possible in that case. And now in blue, you can see the results within our latent equilibrium framework, more or less on par with the vanilla backpropagation. And what is important here now is that we really simulated the time continuous dynamics with a membrane time constant of 20 milliseconds. But each sample, on the other hand, that is each MNIST image, is only presented for a time of 1 millisecond, which is only a small fraction of that membrane time constant. Now next, of course, we can also easily integrate the different deep network topologies, and we demonstrate that by introducing convolutional architectures and trained our networks on both the MNIST and also the more challenging Cypher-10 dataset to obtain again performances that are on par with vanilla backpropagation. And yes, here now there are no tricks involved, such as batch norm or dropout, because we wanted to remain compatible with neurons and microcircuits. And now again, what's important here is that we simulated a network with a full dynamic, so there's no steady-state approximation or phase plasticity involved. Okay. And now, oops. Okay, but there's one thing that I didn't explain to you so far, which is how the errors are being represented in our network, because so far we have assumed that the errors are somehow magically propagated via this dash connection to the apical compartment. But there is an explanation how this can be solved, and it's based on the microcircuit model by Sacramento et al. And this is a complex, detailed model, but the core idea is that the information that is usually represented by the pyramidal neurons is now represented 2 times, first by the pyramidal neurons in red and also by the interneurons in blue. And now first the interneurons, they learn to mimic their pyramidal partners, and then as soon as an error propagates through the network, this error then manifests itself in the apical compartments. And as I already said, in hierarchical networks, this then instantiates a bioplausible variant of backpropagation with real-time dynamics and phase-free continual local learning. Okay. And now to draw a comparison with the original Sacramento model, we did a simple experiment that was solvable for the original microcircuit model without using when using the full dynamics. And as you can see in the upper right plot, the original model is only able to solve this task with presentation times of around 100 times the membrane time constant. But within our model, as you can see in blue, the presentation times basically do not matter. And one can of course also investigate the dependence on the presentation time after 1,000 epochs. And here again, you can see that presentation times basically they do not matter in the case of our model. It only breaks down because the presentation time reached the order of a single time step of our simulation here. And yes, now finally there's one more thing to address here, because so far we have assumed perfectly homogeneous parameters, but instead of using von Neumann computers that evolve the differential equations, we also strive to build low power physical systems that obey our differential equations in silico. However, without the advantages of digitization, of course, we need our algorithm to be robust against diversity. And therefore, we investigated the robustness of our model with respect to spatio-temporal noise. And by spatial noise, we mean, for example, that we use, for example, diverse components such as heterogeneous time constants. Because, for example, it can well be that the prospective mechanism is realized by a different component than the membrane integration. So we differentiate now between membrane and prospective time constants. And now if you model this heterogeneity via multiplicative Gaussian noise on each of them, you can see on the right hand side in orange that our model indeed breaks down if they do not agree. But we came up with a bio-inspired rule that to adapt these time constants. And yeah, as you can see in red now, after adaptation, we obtain still a really diverse system, but can recover the original performance. Okay. And now by temporal noise, on the other hand, we mean, for example, noise on the signal that really transmits the information, which in turn turns the membrane dynamics into a Langevin process. And now this temporal noise has, of course, a different flavor, namely the nature of this noise is indistinguishable from the signal. But we can introduce a low-pass filter with a very small time constant to filter this temporal noise. And yeah, now of course, usually these filtering and membrane time constants are equal and determined by the capacitance and conductances. And as we've seen also, they set the timescale of the physics of the neurons. But within our latent equilibrium framework, however, we can effectively decouple the filtering and membrane time constants. Because using our prospective mechanism, the membrane time constant itself can in principle be arbitrarily big, while not limiting the speed of information processing, whereas the filtering time constant, on the other hand, can be arbitrarily small. And yeah, you can see that on the right-hand side that we can indeed restore the original performance with an adequate choice of synaptic time constant and presentation time. And yeah, this already brings me to the conclusion. So I hope that I was able to illustrate to you the problem that dynamical physical systems of slow components only allow for slow information processing, which then results even in disrupted learning. And that's a problem that's really not negligible and present in a lot of recent implementations of bioplausible backprop. And now, on the other hand, we observe that our brains are not that slow after all. So there must be some kind of mechanisms to overcome the slowness of the neurons. And we provide a solution to this problem within our latent equilibrium framework, where we show that appropriately tuned prospective coding, where the neurons fire an expectation of their future state, can indeed solve this problem. And then we derive the neuronal morphology, the network structure, as well as neuronal and synaptic weight dynamics from a prospective energy function. And in this unifying framework, the neuronal and synaptic dynamics are effectively decoupled, which then allows for continuously active plasticity and also to learn from, in principle, arbitrarily fast-changing input streams. Then further, I demonstrated to you how our theory can be implemented within cortical microcircuits to instantiate a variant of backpropagation. And finally, I showed you the robustness of our model with respect to spatio-temporal substrate imperfections, paving the way for its in silico implementation. And yeah, with that, last but not least, I want to thank all my colleagues and co-workers, namely Benny, Laura, Jacob, Walter, and Mihai, as well as yeah, all the other members of the NeuroTMA and Comp Neural Labs here at the University of Bern. Also the Manfred Steck Foundation and the Human Brain Project for their continuing support. And of course, you for your attention. Thank you.